Did it pop it loose? It did. Go back to my original plan. So my fear is if I knock that thing off, it'll fall down in the crevice.
Okay.
Sure you don't know where you want to put it?
Pretty sure. I don't know. There might be a good spot that we haven't identified just near us.
Roger.
What about the second Osmo spot?
We don't have a second Osmo spot yet.
Okay.
You had suggested that we look for another spot closer,
Yeah.
like farther up the vertical.
Oh.
So that's what we were gonna. No, we decided to deploy the RBR logger in one spot and the first Osmo in the other.
What did I say I had grip force set at? 2?
2.
2 initially.
Grip force 2.
That piece of poly caused problems.
Negative. The poly is no match for a manipulator. The other one is, though.
Yeah, you're going to get 2 samplers right now.
2 for 1. Well, that one works.
Sampled.
Oh, that's a good bottom water sample. Can you note that down?
What number is that?
What number is that?
Can
That
you,
should be
before you
9.
set that down, Dan, can you rotate it so we can see what number
Sure.
it is?
But it should be 0.
Oh.
So it goes, they go from 0 to clockwise around the...
Oh, okay.
So we're going to log that as a bottom sample, correct?
Yep.
Okay.
That's actually quite helpful for priming.
So that was our hair trigger.
I wonder if that's because the nylon rope was in there.
Maybe I'll try grip force 1. I didn't think it would even close with that, though. Appears to. Slowly, slowly.
It's about the speed it was on the last one. You're hung up in that T.
Yeah.
Yes. The first one fired prematurely. 0. Yeah. Mm-hmm.
There goes another little hatchling.
Uh-oh.
I could barely have a hold of it.
And this should be number 2, squeeze number 2.
Yeah, I can see the number from here.
Okay,
That's
cool.
Number 2. You can actually snap a photo. I know it's hard to tell, but you can see the number in there.
Yeah, I take photos of the whole process.
Oh, okay, I'll stop bothering you about that then.
No, you're fine. It's okay to double check.
Okay, video, you can zoom in there. That's good, thank you.
Oops.
I had a reference rock there.
Yeah, you're basically able to kind of go flush in between that octopus and what are eggs. We can zoom in on that exposed egg clutch after if you guys want.
Oh,
Uh-oh.
sticky things.
Taylor Ann, are these getting Nautilus sample IDs?
Yes, they are. And this will be the third
Roger.
003. Yep.
So that's touching there. Happy with that?
Yes, happy with that.
Here we go.
Yes.
3 for 3.
Which sampler number is this?
2.
Okay, Ashley, you can zoom out, please.
And Taylor Ann, the RBR logger and temp logger are just, that's their samples for later.
Right, so we don't record them with a sample ID, but we do notate them in the notes as a special recording. There's 2 different drop downs.
I actually don't want
Yeah,
the pin
and
in the hole.
I got targets dropped for their location.
Okay, cool.
Annie, have we fired a Niskin at this site?
We have not fired a Niskin at this site.
And we should, right?
That was the discussion, yeah. We're going to kind of be at the site for a little while longer, too.
Okay.
Because we still have to place the osmo intake.
Okay,
We still have to place the osmo intake, and we still
next.
have to find another osmo intake place. Okay, so now we
should take that funnel out of the way.
Oh yeah, the funnel. Thank you.
The octopuses may have
This,
accepted it
the
as
porch
their
is
own.
all the way in, right?
What time is it?
Can you see the
What time is it?
stinky baiter with your bubble camera?
Hey, Taylor Ann.
Yep.
On the log sheet, would you mind just writing no funnel used?
Yeah, I did record that in the notes. Yep.
And just make a note that it was attempted.
Attempted, yeah, okay.
Ah. It's a Mongo moment.
Mongo.
Can you get him out without touching the cliff there?
You can't just put
can
them
I
on the
take
front
bubble?
porch?
So I got enough other jewelry there. I could, but then I wouldn't be able to pick up the other stuff. It'd be all over the place. Ashley, can you open the iris there a bit? A little more, a little more. I
Now
can't
Antonella gets to do something.
Finally. Hopefully not destroy anything.
Yeah, if you just bump up on the elbow there, see what happens. Worst that's going to happen is you're going to smack the cliff and the ROV will go spinning crazily across the seabed.
Destroying the whole experiment.
I'm like,
adventure.
if you look
Scattering
at bubble, I'm
squeezers across
right
the land.
on the cliff. Like, I'm already touching it.
Oh, I'm parked on Mongo.
Yeah.
Oh, really?
Looks like I can.
Try do a touch of shoulder to the right. Wrist up a bit. Oh yeah, you are parked on it. Okay, I'll put it on the porch.
Oh.
Sorry.
Were you just going to use the other manipulator arm to hold
That was
the
our
funnel?
original plan, but I'm using it for a parking position now, which is highly illegal. I'll just put this back on top of this data logger, temperature logger thingy here.
Perfect.
Ah, really? Cluttered porch is a sign of a sane mind. Okay, now you want the...
So this is, I guess I'll defer to you and what you think would be easier. We have to find a second place for the additional Osmo intake. We know where we want to put the first one, and we're near the spot, and we're near the Osmo. So would it be best to place that one in and then find a second intake, or would it be best to go find a second intake? And
My feeling is it would be better to find the second intake, because if I'm poking around in there and you already have the instrument deployed, it's likely that we could disturb the first deployment.
Okay, so looking at the screen right now, there's where we measured, and then there's a little yellow sponge on the right side of the screen and an orifice in there with a shrimp near it. Is it easy for you to grab your temperature probe on either the in-situ DOT one or Herc's and stick the probe in that orifice and see if that
Yeah, I think
has
I
any
can reach
readings?
that. Let me do a quick reality check here.
Oh yeah.
Oh yeah,
Okay. Oh.
that's my rock sample.
You made a hatchling.
Did he? Did he? I think they feel a little disturbance in the forest and they run away. Is our porch light on?
Yeah. Yep, it's on.
Roger. Thank you, video. It's actually almost too tight in there.
Get off the probe, lady.
Got a friend joining us here on our probe.
You want to zoom in there, Ashley? Okay.
Well, with our scientific octopus joining us here in our probe, I'd like to say hi to all of the other people joining us on Nautilus Live. We've got people from all over the world: Barbados, Australia, Finland, Luxembourg. Thank you so much for joining in. If you got any questions, send them on in. We'll see what we can do to answer them. Our octopus here, I think they're trying to help us with our science.
Am I far enough in there? You getting anything?
We are getting anything. I was kind of thinking, see under that yellow sponge on the right?
Yeah.
Yeah, there's an orifice right there. If you could almost stick it in there, or the orifice that's like kind of right up in front of you, you can see an octopus in that hole. You can see her mantle and her leg.
Yeah, there seems to be some little white things growing around where it's coming out there.
Yeah, so just we're trying to get as much
Yeah.
of the flow
Can
as
you,
possible.
zoom out there? Actually, I have to reorient the manipulator here and
Okay.
or the probe. Okay, can zoom back in. Yeah, zoom out just a touch. Sorry, I don't have any more right on the camera. Can open that iris right up. There we go. Just trying to see the tip of the probe there. Okay, that's pretty much in the hole.
Just hold it there for a second, please.
Hold it.
See a little movement on the oxygen coming down. And maybe the temperature coming up slowly.
If you zoom out a little bit, and then
It's going up.
it's going up.
Yeah. Temperature's up at about almost 6 degrees. And the oxygen, I didn't see where it was before we started moving it, but now we're at probably about 85 micromolar.
Okay. Dan, do you see that hole in the very
millimolar.
top of your screen?
Just above the bright yellow
Yeah.
sponge.
Top center.
That, and then also top center. If either of those two holes get higher readings,
Trying to Spider-Man you.
Let go of me.
yeah, she's
She's-
just really eager to help.
She's our up-and-coming scientist octopus.
Oh, she's gonna be along for the ride. Keep going up.
Yeah,
Yep.
so right in this area, right where there's an octopus in that crack, and I think there's a hole right behind you. Yeah, like right next to her.
Where the shrimp are.
Yeah, yes.
I see it.
Exactly.
We have a stick of pointing if it helps
Okay.
for future reference.
Thank you.
Is that iris all the way open there?
Ooh, there goes a little fish.
Yeah. Sorry, it's gonna blow it out a bit, but I just want to see the tip. I think so. Is that the hole you're on about?
Yes. Thank you.
Okay, Ashley, you can shut it back down a little.
Do you want me to zoom in?
Sure.
Can you iris down and get a nice shot here?
What's our temperature at?
We're seeing a little drop, but that might be a lag.
I think there's a lag to it.
Okay, now it's coming back up.
Okay.
It's around 6-ish.
That's, and then the oxygen, like 70, 85. Ooh, that was a good jump.
Scale just changed. 85-ish.
Okay. I think this all seems to be pretty consistent in this area. We could explore up a little and see if we can find another hole. I'm comfortable deploying both intakes here.
Yeah, I think this spot's good.
Okay. So Dan, now what we want to do is we want to deploy the temperature logger, probably the yellow one since it's already taken off.
Okay.
And then go back down to the Osmo sampler and put the blue handle in the first temperature logger position and then the green handle in this one.
Okay, I see. You want to zoom out there for me? While I'm here, if you want, we can poke along that crack on the way down and see if you get a higher temp.
Yep, that's good.
Yeah, you can zoom back in there, Ashley. It's looking kind of for the bacterial stuff growing there. Anything there?
Give it a moment. It has a bit of a latency to it.
Roger.
So I think this is about the same as what we were seeing up above.
Okay.
Around 6 degrees. Thanks for that.
Moving on.
Looks like you're almost oh. Nope. I was gonna say the octopus almost had let go.
Yeah, I was kind of hoping she would
I think
grab the rock instead of.
she's in the process of it.
If you, oh, you're stuck to her. She's stuck to the rock, huh?
Let's just pause. It looks like she's taking her arms off. She just needs a moment.
While we're sitting here, is it possible to zoom in on those eggs and get a nice shot of them?
Just happened to be front and center.
Oh, those are her eggs. Look at those eye
They
spots.
are, yeah.
All those little black dots that look like pieces of pepper, those are eye spots. And that white coloration.
There's hatching on the right.
Oh, you see a hatching one?
It looks like on the
Over
far
there.
right, there's one that's hatching out right now.
Ooh.
Oh, yeah.
Ooh.
Okay.
Wow.
And possibly, oh yeah, one on the bottom left as well. I think that might be an arm reaching out of the case.
Oh, yeah.
2 arms now.
Yeah.
It's awesome.
Oh, it's like that scene in Jurassic Park when the baby velociraptors are hatching.
So cool.
Life will find a way.
Tiny predators.
There they go. So you were talking about the eye spots. So the eye spots are telling us that these eggs are indeed have baby octopus in them. And is it
Yes.
telling
Ooh.
us that they're pretty well developed, or will you see that at any stage?
No, you won't see this at any stage. The newer eggs, so if you look actually on the right, that's a really good contrast. You can see those eggs are much more clear and shiny and kind of translucent. Those eggs are either really new or they're not. They don't have any embryonic development in them.
A lot thinner.
Yep. So these eggs that we're looking at now are, well, as we're witnessing, they are ready to hatch. And whether that's related to the fact that we're actually in this area working isn't something we can necessarily draw a conclusion on today. But do you notice that shrimp who's in there investigating the eggs? He might be trying to scavenge.
Grab a bite to eat or so?
Yeah, that's what we saw at the other site of these guys, of this type of octopus where they clustered. The shrimp seem to be highly interested in the spent egg clutches.
Seems like a great source of nutrients.
So when a mother is actually brooding on them, she's not providing any more nutrients, but she's protecting them from predators. What else is she doing to provide protection?
She's basically protecting them physically, and then she jostles them, so she keeps them oxygenated.
Mm-hmm.
As far as I understand, that's
So those are the two
about
main
what
things.
they do. Yep.
Going along with that, we had a question of why do octopus eggs need to be vented? So she's sitting there venting them, protecting them. Is that to help them grow? What?
Yeah, they intake oxygen through their core. Their shell's called a corion, and then that's where the oxygen enters. And so if you have, you know, one of the eggs at the bottom of the clutches that's kind of squished against the rock, it might not get as much opportunity to see that oxygen as some of the outer egg
the one on the
clutches.
left just
Oh.
is hatching.
Look at those little arms coming out.
Free, get away from that shrimp.
Uh-oh, shrimp.
Run.
Oh, this could be so cool.
Run away. Oh, octopus shrimp battle. Go, baby, go.
Go.
Are we filming this?
Oh, yes.
Yeah. Oh yeah.
So that
You
one
can see
still
the
has a
yolk.
sac.
Mm-hmm. So that's yolk provided by the mother, and it has nutrients to help it further develop.
Oh. Oh. Come on, little guy.
Wow. Wow.
The shrimp had to eat too.
Mm-hmm.
Did he get it? Oh.
This is this
down.
complex biological
There you go.
environments. You have
that?
interactions.
If you zoom out just a touch, it'll. There you go.
And I kind of feel like a baby octopus could take him.
Literally his first minute in the world, and this is his trouble.
It's a tough life as an octopus.
Well, the wild is a rough place.
It is.
This makes me think of sea turtle nests and hatching, and how when sea turtles are hatched, they have to make it free from all the predators that are on land and then get into the water where there are predators waiting there as well. And here, the octopuses, you know, we saw the other ones hatching and they swam up first. So, I mean, they're contending with shrimp and who knows what other predators down at the seafloor right when they come out.
How's that oxygen on the probe doing?
Oh.
Did she leave?
No, she's still there.
The oxygen is at about 80 micromoles and the temperature is at 6.4 degrees.
I think I meant to say the octopus on the probe,
Uh-huh.
but thank you.
So now I'm not sure who's swimming, the
Yay!
octopus or the shrimp. There he goes.
Who wins? Octopus.
Right now the octopus, but the shrimp is at it again.
Shrimp will just sit and wait.
Shrimp didn't
Oh,
get
he
his meal.
knows.
He's hungry.
And he's still
He's
hungry.
angry.
Wonder if he can puncture those egg capsules.
Oh, that would be interesting.
Okay, that was really, really cool. Should we keep probing for temperature and oxygen and try to get this Osmo sampler deployed?
Sounds like a good idea.
Sure, I can do
Temperature's up to 6.8.
one more probe here with my.
Are we in a hole?
We're just hovering above it.
And it's at 6.8?
You can see in the bubble cam there.
Yeah.
It's not possible to go in that hole, is it?
Yeah, it's down in the hole a little more.
What's it counting up to?
Well, right now it's still at 6.8, but we'll give it a moment.
Can push in past the cable and the rope there, Ashley?
Sorry, you want me to push in?
Yeah, just past that. So the, that didn't help. Just so the rope's not in the shot. Zoom in on the shrimp a little. Is that full zoom,
I'm
is it?
full zoom.
Looks
Sorry.
like it's about 6.4 degrees here.
Okay, that's good, Dan. Thank you.
Okay.
So while our
Can't really see
shrimp
what I'm doing
here is trying to grab a snack, we had a question about the octopus eating while they're brooding. So they're not eating while they're brooding, but scientists, could you tell us where that energy is coming from? How are they able to survive here for so long without eating?
Well, octopuses are senescent. They only have one clutch, and after they have that clutch, their mission is to protect it, and so they put all of their energy, their stored energy, towards that. And the octopuses themselves, the octopuses senesce. They're superfluous, which means they only lay one clutch in their lifetime. At least that's what's consistent with other octopus species, and we suspect is the case with these guys. And so after they lay their clutch, all of their energy goes towards protecting the eggs, and so they don't intake new energy.
So everything they have left goes to their babies.
Yeah. And the word I was looking for was semi-pearlous.
Semi-pearless, and that means they only have one clutch in their lifetime?
Yeah, I'm probably
Gotcha.
mispronouncing it.
Semoparis?
Yes.
I have a
Yes.
request to, if Science, you could talk a little bit louder. It's a little hard for our audience to hear you.
How's this?
That
Better.
sounds great.
Oh,
to
hello.
me.
So we still have our hitchhiker there, and I still have that instrument in my hand.
Well, can you just put the hitchhiker on the instrument on the basket? The hitchhiker will probably leave.
Can do. Yeah, you wanna move on now?
Yeah, let's place the Osmos.
Okay. You wanna zoom out for me, video?
Please.
Thank you.
Nautilus is taking new interns in the form of octopuses.
Very competitive internship.
It's the deep sea exploration internship.
So now that we've determined this is a good place for equipment deployment, we're going to put out the Osmo sampler. Is that right? All right.
And then we could, your Niskin too, which you probably didn't forget about.
Right.
And confirming we're doing the yellow temperature sampler here.
Yeah, so the yellow temperature logger will go in that hole. With the green handle.
They really are small. I thought they were like basketball size.
They're more like grapefruits.
Yeah.
Yeah.
They're just little guys.
So we've got a question that I think is an interesting discussion point for you guys, Science, if you have a second. Would all these octopus have arrived at the same time? Do they trickle in over different years? Do we have any idea of, are they showing up? Are they leaving? What's our thoughts on that?
You wanna push the porch
Our
out a little?
thoughts are that
Stop
they
when
are
it hits the
coming
cliff.
not all at the same time. And our thoughts are that because it's not been really... You'll see different stages of egg development at this site alone,
The
which
vehicle
suggests
moves,
that eggs are being
Oh yeah, that's it. Sorry.
Yeah, that's it.
You're good. You're good.
Laid.
released, laid at various times for the octopuses. Right, so even when we were just zooming in 15 minutes ago and we had that really well-developed clutch on the left, and we compared it to that more translucent clutch on the right, so that suggests that the octopus that laid the eggs on the left has probably been there longer, thus coming before the octopus on the right. But honestly, we don't actually know for sure. We just have evidence that leads us to believe that they don't all come at the same time.
Sorry to interrupt, guys. Do you want the yellow one, or can I do the green one?
You can do whatever works easiest for you, and we'll just take good notes.
If they trickle in at different points, do we have any idea if it's sort of a ongoing thing all the time? Is it seasonal? Do we know if there's any effects on it?
So at this site, I've asked the scientists who have visited multiple times, and I've gotten the answer, and you guys can back me up, those of you who have been here multiple times, that the octopus population doesn't really seem to change, right, between years
Well,
or between visits.
go ahead, Amanda.
Well, we've been here 4 times total, and there have been octopuses at all 4 of those times. And so there are octopuses present in, let's see, October, March, April, and August. So we know that at least those times of the year there are seemingly thousands of octopuses here. But we don't know the sort of immigration rate of new octopuses or the
Happy-
immigration
That's
rate
great
or
data.
departure
Yeah,
of
that's great.
octopuses.
Opening.
But it does seem, yeah, like
Actually, could you stick the end a little more into that hole?
Yeah,
Unless
can
that's
do.
just shadow that I see.
we have, I mean, this site was just discovered a year ago, so we have 1 year's worth of intermittent observations. And from that, it looks like it's a persistent feature here. It's not just like this is a aggregation that showed up and will eventually disappear again.
Yes, bury it in rocks. Perfect. Keep it in place. I was kidding.
Landslide.
You got
The
it.
rocks are really
Can
friable
we
here.
find
Can
it
we
again?
maybe
Pretty
just move
friable.
that top rock off of it, and then we can just drop a marker?
Yeah, is this a temperature log or
This is a
the
temperature
osmo?
logger. And
Would you
if
like a
put
rock
green-
sample?
Why not? If you have it,
I do.
yeah. It's a relatively
When's our
decayed, and
second temperature
maybe we
logger?
can look at some.
It definitely came out of the. I'm gonna zoom in there a bit, Ashley, and we'll take a picture of it.
Oh, great. That's got a lot of those worm tubes on it. We can see what worms those are.
I think it's a hard rock. It was just cracked there.
Great. Do you have any more zoom and possibility to turn the iris down so we can see detail
Yeah.
on those tubes?
We do.
Any more iris down? Just a little. Okay. Well, so they're tubes made of what looks like calcium carbonate, so hard tubes. Beyond that, it doesn't look like it has any surface kind of architecture to it. Great, thank you.
Is that like a class of tube with the calcareous casing?
Yeah, there several families can make calcareous tubes, and then there are other families that would make tubes out of different materials. So you can kind of
Probably
narrow down
Put it on that ledge there.
What's
to
that?
You can probably
a
put it
couple
on
of
the ledge.
families
No, that's
based
a
on the tubes.
collectible sample going in the box.
Oh, really?
Oh yeah. Okay.
Look at that. The octocoral like curled up itself like a vase up in that top right corner.
Yeah, I wish I knew what that was.
We've put feelers out to see if the world knew, because we've seen it with the other site that has the octopuses too.
You can.
They have 8 tentacles. Like
Starboard?
someone referred to it as a snardiator.
No, I don't need that. Let's look at the octopus.
All right, and what
The other
box are you
camera
planning
will
on
do.
putting that sample in?
Box E
Okay.
or F.
One camera, Dan. Nice.
Yeah, you can switch to the other camera if you want.
Which camera do you want?
Okay,
And that went into box E?
correct.
Okay, cool.
Sample 4.
Yep, sample 4.
Rock. Rock, rock. Oh, watch out for that cliff. Video, you want to zoom in there and see if that temperature probe's buried or what's the situation
There's
there?
the handle. The rest of it's buried.
I'd say it's in the shimmering water.
Take a grab of that, please.
Happy with that, Science?
I'm happy with that.
Okay.
Do we have a grab of that? Okay, great.
Wow, that's great. That's really great.
So that's our 2 victims for the Osmo, right?
And it worked out that the colors are going to go well. So if you could put the green Osmo handle with the green locker.
You're pushing your luck.
Hey, the squeezers worked, so I feel very lucky right now.
Oh, I will give it 110%.
What?
Okay, if you want to zoom out, we'll go victimize the Osmo.
I thought it looked like Anthomastus. His is a cousin, right?
Mhm.
So,
See. Right.
We had a question
Bathyalcon
here
robustum.
of whether we know that if these baby octopus come back to brood here,
Uh-huh.
just as the same way turtles would go back to the same place they were born. Do we have
Tultree's
any
out just
indication
slightly
that octopus would
more.
come back?
I would say we don't have any kind of evidence what to say one way or another, whether it's the same octopuses that hatched here that then come back here. We just don't have a way to track that right now, and with only having been here for a year, we don't know that yet. A lot of animals do have various forms of homing, so a lot of animals have reception or sensation or sensory abilities for the magnetic field. So it is possible that they could navigate here from a lot of places. One thing we do know is that this octopus species we don't normally see in these numbers, which is what makes this place so neat. But it means that normally we would just see one of these, and then maybe, you know, a whole dive we would see one of these octopuses, if we see any at all. So they must be coming from pretty far away to this site.
It looks like you're on the craft arm.
There's a tunicate
Who dat?
on the right.
Oh, that's the
Culeolus.
That's Culeolus.
Yes, that stalked, clear-looking dog head. Looks like Snoopy almost.
Can you look at the craft of the bubble?
Does.
camera?
And what was the scientific name of that tunicate?
Culeolus berryi, named after Jim Barry.
Well, I don't know if it's berryi,
Oh, no?
but it is the
There are a
genus.
couple others. Okay.
Yeah.
Culeolus. C-U-L-E-O.
Am I sitting on that rock?
C-U-L-E. Sorry? Let me write it down.
C-U-L-U.
Culeolus. There we go. C-U-L-E-O-L-U-S.
Great.
And that large white sea anemone that we've been seeing, we've identified now from Mbari's Deep Sea Guide. It matches with Bathyalcon robustum. Ooh, with the alternate name, Anthomastus robustus.
Yeah, so
Ah,
the genus changed.
got it.
So let's hope when we pull these off that.
So the green one was the one all the way up the hill, right?
Yes.
Correct.
All right, I haven't seen this deployed yet.
Yeah.
So
Is
we're gonna
Mongo
pull
still
those out.
stuck? Can Mongo hold the basket down when you pull that up? Not the basket, the crate. The milk crate.
No.
Oh, sweet.
Maybe. Is that a rubber band lifting that basket up?
Yep.
Cool.
So this tether you said is about 4 meters, is that right?
You know what? That tether is probably also, I don't think it's zip tied. I think those are rubber bands too.
Hope those pop off.
Yeah, it's supposed to come off.
Hope you did over under.
So we should probably upright the crate.
Can you reach with Mongo? Why don't you get Mongo out? You can use the
Okay.
bubble cam.
Thank you. Help if I'm able to keep them.
Mongo to the rescue! You,
So we had it placed on the ledge, kind of where the
yeah, I didn't like it there.
Okay.
I'm joking. It's we'll probably set it back down there. So I'll see if I can get the hoses deployed.
Great.
But we may want to move the crate up closer. So
Yeah, we could put the crate at the top and then have the hoses going down.
yeah, that way we could see the whole mess while because we're gonna lose visibility of it here as I move up the slope.
Okay.
But we're kind of committed now that I've started deploying the hoses. So if you wrist right there, so I can see the jump, and then open. I'll just put it in your jaw.
Want me to just right some more?
What's that?
Never mind.
Okay, close.
Yes.
Got it?
Got it.
Letting go. Okay, you want to bring Mongo's shoulder up a bit. Perfect. Okay, hold what you got there. Video, can you push in there a bit? That's good, thank you.
That octopus below is just like, if I stay here and I don't move, maybe they won't see me.
Appears to be another one there.
That white? I thought it might be a nude break. No. No, maybe not. It's not moving.
Really?
The gray one? Mm-hmm.
We only buy the best rubber bands in our lab.
These are extremely robust rubber bands. My mom's cats would love these. They're rubber band thieves.
I support that. Rubber bands.
It's pretty adorable. There it goes. Yay.
Hey.
That octopus isn't even bothered at all by this. Are you supposed
Oh, yeah. So, forgot you have the basket. Okay, you can zoom out, thanks. Okay.
Okay. Very good. Okay. It was kind of up on that ledge up under your predator arm.
Yeah, I'm gonna set it down in the crevice for now.
Oh, sweet. If that'll reach and that stays steady.
Looks like a good spot.
I could put it on the ledge, but it's probably gonna slide off when I pick up the hose and fly up.
Okay. Let's not do that then.
Is that where you want it, on the ledge?
No, not if it's going to fall off when you pick up the hose. I just want it somewhere steady and where the hoses are going to reach to. That's really the only stipulations.
Yeah, we'll have to pick it up again to get the other one off, I'm sure.
Okay.
So it might drag around a bit, but that should be all right. Everything's in the milk crate, right? Is that true?
Yep. Yeah, and the hoses, they do have a little pull because the capillary tubes are protected by that external green hose that you see. So you should be good, although we won't know until we really collect it.
So you designed it to be ROV pilot resistant, is that what you're saying?
They designed it to be ROV pilot resistant, I hope, is what I'm saying.
Don't go too far because we're going to need you again.
Okay. Yay.
But you can tuck it in behind the porch there.
Yeah. You know, it's so neat. We keep looking at the spot from different views, and it always looks so drastically different. That's great. I think I can see it right below you. We went past it.
I wonder if I could have reached. I don't think so. But oh.
Fun fact, since we see all these sea stars here, these are called Brisingid sea stars, and that particular group has given up the
I wanna show
predatory
me the
way of
crevice
many
with
sea
the
stars. And they are suspension feeders. So that's why their arms are sticking up into the water column like that instead of being flat on the seafloor like you'd expect with a lot of other sea stars.
That one looks like it's got a bazillion arms, but maybe it's two.
It does.
So
like
this
Medusa.
one, we're going to want to stick in the same hole that we stuck the green temperature logger in. And so as we were scrolling up, we zoomed over it a little bit.
I believe is right below me. Tell you what, I'm going to put it here where I can see it for now, and I'm going to go get the other one and bring it up here, and then we'll get in a position where we can see both of them, and we'll do a final placement.
Okay.
Get off me. That's the little yellow sponge right there, right?
Yeah, that's where
So
we placed it.
It's close. I'm afraid if we take the time to place it now, then and we move the crate around, we'll come back and have to remove it anyway. So if I get the other one up there, then we'll put the green one last.
Okay, thank you.
Do we still have our octopus friend on the front porch?
What's that?
Do we still have our octopus friend on the front porch?
I don't know.
Yes.
She's hanging out. Ah.
I think I used my previous landing spot, which worked out okay.
Thanks.
Sorry, watch out, Patty Panties.
Oh.
Whoops. I was checking things off here.
Oh, you got,
So
okay.
same drill,
Double
hand it to
duplications.
Mongo again.
Okay.
But you can confirm.
We've got the same.
Why is there blue, green, and red tape on the arm?
That's when I'll let Aaron explain that one.
When we're doing the
Sneak it
white
in on me.
balancing and the black balancing, it's a color reference for us.
Oh, okay. Thank you.
I did sneak in.
Does that have anything to do with all of our probes having colorful tape, or is the colorful tape just to keep
No.
them
Ready
separated?
for it?
No, it has. The colorful tape's always on the arm.
Gotcha.
The samplers just have colorful tape so that we can, one, because that's how this particular science likes to color code a science for easy identification
Where are you?
and sample naming. But also it kind of sticks out on the seafloor, hopefully.
Got it?
Got it. Do you want me to move it up?
Yeah, you come up a little. So it spins. That'll do. Okay, set it down a little bit. More. Or swing it into the porch.
Okay.
Yeah, it went.
Do you
Oh,
want it into the porch?
Yes.
Okay, hang on.
Hold on, I'll give you some help. Hold on.
You got it?
Yeah, swing right a little more.
Okay.
Or put the porch out, one or the other.
Are you putting it back on the porch?
No, I just want to, well, I wanted to rest it there so I could get the T-handle.
Okay. You want me to, okay. Oops.
Okay, hold what you got there. Can you come up a little? Or left a little?
Oops.
That should do it. Okay, hold it. This is not working.
Octopus over on the bottom right is trying to investigate the arm.